Welcome back. Okay, here we are. This is day 2 of our epigenetics lecture. And if you'll remember from last time, I asked you to just go ahead and take notes, quote, the old-fashioned way, unquote, on notebook paper or on your Surface. And what we're going to do today is we're going to go ahead and revisit the mystery of the puzzling pedigree of the starbelly sneetches and see if we can explain the mode of inheritance in this population. So I think what we ought to do though is this. Let's back up. Let's back up a little bit here. Okay, to this slide. And what I'd like for you to do is take out a blank sheet of paper and draw this pair of chromosomes on your blank sheet of paper and label this normal. And so what we're looking at here is a normal pair of chromosome 15s, the blue one from the person's dad, the pink one from the person's mom. And again, we can see here that this individual's mother imprinted the S allele before she passed that pink chromosome on to the child. And on the left-hand side, dad imprinted the U allele on his chromosome 15 before passing it on to the child. So this is what every person in this class in real life has. This is what your chromosome pair number 15 looks like. So let's go ahead and pause the video and just go ahead and draw this pair of chromosomes with the SS gene pair on them and the UU gene pair and the U imprinted on the chromosome that came from dad and the S imprinted on the chromosome that came from mom. So go ahead and draw this pair of chromosomes. Pause the video and do that, and then come on back. Okay, now that you're back, let's do this. Let's look at a situation where this portion of chromosome 15 from dad has been deleted. And so what we have here is an embryo. Again, the embryo that owns this pair of chromosomes would have no functioning S allele at all, and that results in a condition called Prader-Willi syndrome. So go ahead on another blank sheet of paper and draw this pair of chromosomes the way they look. And go ahead and pause the video while you do that. Okay, now then, let's continue on. Now let's look at the other situation where that same region of chromosome 15, only this time the one that came from mom, is deleted, leaving the embryo, the child, the person who owns this pair of chromosome 15s with a situation where he or she has no functioning U allele, resulting in Angelman syndrome. So again, go ahead on another sheet of paper and draw this situation, this pair of chromosomes that represents what we would have with Angelman syndrome. So go ahead and pause to do that and then come on back. All right, so now keep your 3 diagrams, your 3 pictures handy. And what we're going to do now is we're going to transition back to the mythological starbelly sneetches. And so what I'm going to do is I'm going to point to a sneetch in this pedigree chart. And when I point to the particular individual, I want you to, since you can't really hold these pictures up to show me because we're not in class together, just go ahead on your own. Of the 3 diagrams that you've just made, when I point to a sneetch, then you go ahead and point for your own knowledge, your own edification, your own reviewing. Point to the diagram that that particular sneetch would have. Point to the diagram that shows the chromosomes that that particular sneetch would have. So, what we're doing here is we're saying that blue stars, blue sneetches, that condition is analogous to Prader-Willi syndrome in humans, and red sneetches, or sneetches with red stars, those sneetches are analogous to humans who have Angelman syndrome. And then the unshaded pedigree symbols represent normal, that is, sneetches with no stars on their bellies. So let's go ahead and get started and see if you can make the connection here, and it will help you, I think, to understand what in the world is going on with these sneetches. So here we go. There's the first sneetch I'd like for us to look at. So identify which of your 3 drawings of chromosome pair 15s, which of your 3 would be the one diagram that would represent the chromosomes that this particular sneetch would have. So go ahead and do that. You can even pause this video if you need to, if you want to. Okay, if you said this, then you are absolutely right. You should pat yourself on the back. Okay, so yeah, there's a normal sneetch with 1 functioning S allele, 1 functioning U allele. That's what you're supposed to have, because the SS gene pair, remember, is supposed to be monoallelic, and the UU gene pair is supposed to be monoallelic. And in humans, those 2 gene pairs are 2 of the approximately 200 gene pairs that are imprinted this way in humans that work in a monoallelic fashion. Okay, let's look at the next one. How about this sneetch right here? So go ahead and identify among your 3 drawings which pair of chromosomes this particular sneetch would have. Now, if you said this, then boom, you're right. Excellent. So this individual has only 1 functioning U allele. The broken chromosome came from his father, so he is a sneetch with a blue star on his belly. That's kind of analogous to Prader-Willi syndrome. Okay, moving along. Keep track of your score here to see how you're doing. How about this particular sneetch right here? Go ahead and identify among your 3 diagrams the chromosome pair that this particular sneetch would have. All right, if you said this, then you should pat yourself on the back again. Awesome. Okay, and now how about this one? This is a female with a blue star. And there's her chromosome pair number 15. Okay, so here's the question that I have for you. I want you to think about. Who did she inherit her broken blue chromosome from? So go ahead and pause the video if you need to. Think about that. And then we'll come back and compare answers. If you said she got it from her dad, you are absolutely right. Because remember, dad's going to paint his chromosome 15 blue before passing it on. Now you realize, I'm sure, that in humans, we guys don't really paint our chromosomes blue before we pass them on. It's just a way of envisioning the process of a guy stripping off the epigenetic markers or the imprints that came from his mom and putting his own male imprints on before passing them on to his child. And that's what's happening here. All right, how about this niche right here? This blue starred female. Go ahead and identify chromosome pair 15 diagram that would represent her chromosome pair 15. Is this pretty easy? And if you said that, then you're absolutely right. Here we go. How about, let's go on to this one. How about this niche right here? She has a red star. And my dog is out there barking at deer in the field behind the house. I don't know if you can hear her or not. Yeah, you probably can. Okay. Go ahead and identify the diagram that represents this niche's chromosome pair number 15. All right. Is this what you had? If so, very good. Question. This is analogous to Angelman syndrome. Who did she get her broken pink chromosome from? And again, if you said she got her broken pink chromosome from her her blue mother, you're absolutely right. And now here's a classic case here of where her blue mama, who inherited a broken blue chromosome from her dad, mom stripped off the epigenetic markers off that broken blue chromosome, and before passing it on to her daughter, put on her own pink markers before passing it on. So it's almost like you strip the paint off and you put your own paint on before passing them on to your kids. Now let's look at this male here, this red starred male. Go ahead and identify the diagram that represents his chromosome pair number 15. Okay, is this what you thought? If so, again, you should pat yourself on the back. Are you 100% so far on this? If so, you should party or something. Although it's kind of hard with social distancing. That's kind of a bummer. So anyway, let's move right along. And how about this niche right here? Okay, it's a female with a red star on her belly, and go ahead and figure out which diagram matches hers. Is that what you said? Very good. Okay, analogous again to Angelman syndrome. So in humans, if the broken chromosome 15 comes from mama, then in humans you get Angelman syndrome. In starbelly sneetches, you get a red star. Okay, how about this niche right here? All right, if that's what you thought, then again, you're right. And how about, this is the last one we're going to do, this female with a blue star. And there we go. So notice here, she got a broken blue chromosome because it came from her dad, who happened to have a red star. But again, before her red starred father gave her that broken chromosome, he stripped off the pink paint and put his own blue paint on. I hope this kind of makes sense a little bit, but I wanted you to get a taste of this relatively new area in genetics called epigenetics. And the more we're learning about epigenetics, the more we're learning that we thought we knew everything about genetics, and now we realize we don't. uh, when I first learned about epigenetics, I thought to myself, self, I thought I understood genetics, but then now epigenetics just raises a whole new series of questions and teaches us some humility, I think. Teaches us that all those things we thought were hard and true facts in genetics, there are exceptions, and this is one of those strange examples. So there's a lot to be learned in the world of epigenetics, and this is just a taste. What we've looked at here with red and blue stars and Sneetches and the connection between Prader-Willi syndrome and Angelman syndrome in humans, that's just one song. If you'll want to think of it that way, that's just one song in uh, a whole fugue of a whole bunch of songs that are part of the whole world of epigenetics. And so you're going to learn more about this world of epigenetics in our next lesson. The next time we're together, I'm going to have you go to a pretty cool interactive website to learn even more about this fascinating field of epigenetics. But I think this is a good time to end, and this is actually the end of this epigenetics lecture, this 2-day lecture, which is just designed to give you a taste of epigenetics. And like I said, we'll go into more depth next time when we go to the interactive website. So, have a nice day. We'll talk to you next time.